Hello, my name is Mindy, and I'll be presenting my project on the role of nkx3.1 in sclerotome development. So this is a picture of a human embryo at around 26 days old, and along its back we can see these repeating structures which are called somites, and they appear very briefly during development. Now if we were to zoom in on a cross-section of one of these somites, we would see something like this, and we can see that each somite has three compartments. So the myotome later gives rise to our muscle, the dermatome later gives rise to our skin, and the sclerotome later gives rise to our vertebrae, among other things. And so understanding how somites develop into these structures is important because it helps us to better inform treatments for embryonic defects. So since this process is difficult to study in human embryos, I used Zebrafish as a model. And on the left here we can see a picture of a zebrafish embryo, and on the right we can see the somites in zebrafish. And so zebrafish actually have the same three compartments as humans, but they're just arranged a little bit differently. So in this green region called the sclerotome, early in development, cells will migrate from the top and bottom compartments toward the middle, where they then differentiate into different types of cells. And this migration is very important for these cells to differentiate correctly. So all sclerotome cells express a gene called nkx3.1. And on the left here, all of the dark purple staining shows nkx3.1 expressing cells in a zebrafish embryo cross-section. And then on the right, we see those same cells, and we can see them during their migration in these nice diagonal patterns. But why is nkx3.1 important? While it's well studied in contexts like prostate cancer, its role in embryo development isn't very well known, so my project aims to uncover more knowledge in this area of research. And so one study actually showed that the loss of nkx3.1 in mice leads to vertebral defects, where the vertebrae fail to close properly, as you can see on the right. And since migration of sclerotome cells is critical for vertebral development, I wanted to see what the role of this gene was in the process of cell migration. And to test this, I looked at what happens to these embryos if we take out the gene nkx3.1. So our lab has a mutant zebrafish line without this gene, and so I performed in situ hybridization with our embryos. And so this is an experiment which uses complementary probes that'll bind to their target mRNAs and will form a purple stain. So essentially, all of the mRNAs that we want to look at is going to be stained purple, and we can see their localization in the embryo. And since nkx3.1 is expressed in all sclerotome cells, naturally I use this nkx3.1 probe to look at all of the cells in the sclerotome. And so on the very left and in the middle, we can see the wild type and the heterozygous embryos, and we can see the cells starting to migrate from the bottom. However, in the mutant on the far right, we actually didn't find any migration happening. And this was a pretty surprising finding, and it suggests to us that nkx3.1 is regulating sclerotome cell movement somehow. And I confirmed these results using another probe, which essentially labels the same cells, and we saw the same result as we would expect to. And so next I wanted to see, so if these embryos are unable to undergo proper cell migration early on, I wanted to see if it was affecting their development later on. So I allowed these embryos to grow for about another day, and then I stained for two cell populations that are derived from the migrating sclerotome cells, the fin and the tendon precursor cells. And surprisingly, both of them appeared completely normal, suggesting that these embryos are somehow recovering from the initial migration defect to be able to continue to develop normally. So to conclude, it seems that in the presence of nkx3.1, cell migration is able to occur normally. However, if we take out this gene, cell migration does not occur. But even without it, we saw those embryos that were able to somehow recover and continue to develop normally. So moving forward, it would be interesting to study how these embryos are recovering from the initial migration defect. For example, we can see if there are other genes that are compensating for a loss of nkx3.1. And also it'd be interesting to use time-lapse imaging to be able to track cell movements directly in these mutant embryos in real time to see what's actually happening to those cells that are unable to migrate. And so this brings me to the end of my presentation. So I would like to thank the Huang Lab for their support and their guidance with this project, as well as the Aberrant Center for funding my research. And thank you for listening.